Calm, yeah, so it's all the topic 5 sheets patterned there. Obviously, man can't show the sheet in it because of copyright, yeah, but obviously follow it, go to the description, look at the thing, yeah, and just follow the whole thing, yeah, pattern it, you'll be calm with the whole topic, yeah. So not. Alright, cool. This is the first sheet inside topic 5, yeah. The sheet is called photosynthesis, yeah, so I'm gonna go through it now. Alright, cool. Question 1A, yeah. Question 1A says, in photosynthesis, which chemicals are needed for the light-dependent reaction, yeah? So obviously the answer is gonna be the bottom one, yeah? NADP, ADP, PI, and water, yeah? So NADP, yeah? So NADP is obviously gonna be used in the light-dependent reaction, yeah? It's gonna be the electron acceptor, and it's gonna accept the H plus as well to form NADPH, yeah? Calm. Then also ADP, yeah, and PI. Obviously that's gonna form ATP, yeah, which is gonna be needed in the light-dependent reaction as well. It's gonna be made in light dependent, yeah, needed in light independent, yeah. Alright, cool. And then finally, yeah, water, yeah. Obviously that's just bait, yeah, it's water and you need it for photosynthesis, yeah. Alright, cool. Question B, yeah. Describe what happens during photoionization in the light dependent reaction, yeah. So in photoionization means that the electrons absorb light energy, yeah, and get ionized, yeah. So what's happening is the chlorophyll absorbs, yeah, the light and loses electrons. Yeah, the electrons become excited, yeah, and they leave from the chlorophyll, yeah, they lose electrons, yeah. Calm. That's all you need to write. That's 2 marks, yeah. Alright, calm. Question C, yeah. Explain why the student marked the origin line using pencil instead of ink, yeah. So obviously in pencil, the origin line stays in the same place, yeah. So obviously if it was drawn in ink here, it would start moving up the thing, yeah. You get. So obviously it stays in the same position, same place here when it's in pencil, yeah. Alright, calm. Question D. Describe the method the student used to separate the pigments after the solution of pigments had been applied to the origin, yeah? So what he's basically asking here, yeah, it's a bit ambiguous here. What he's trying to say is that um what's he gonna do, yeah? Like to separate the pigments. Obviously he's gonna put it in a solvent, yeah? And you gotta say that he's gonna put it in a solvent that's below the origin line, yeah? And you gotta say that it's that he takes it out before it reaches the top, basically. That's what he's asking, yeah? So the solvent level is below the origin line, yeah? And the paper is removed there before the solvent front reaches the top here. Go on. Question E here. Calculate the RF values here of the pigments that can help to identify each pigment. Yes, you got pigment A, which is RF 0.95 here. So remember, RF here is equal to pigment here. So distance that the pigment traveled here divided by distance that the solvent traveled here. Calm. So what you need to do here, yeah, is you need to find the distance that the solvent traveled here up the paper, yeah. So what you want to do, yeah, is you know that the RF for A is 0.95, yeah. So you know that's the RF for number A, yeah? And now what you need to do is find the solvent, yeah? So you need to rearrange the formula to find the distance that solvent traveled, yeah? So using your ruler, yeah, measure how far up part A moved, yeah? And that should be about 5 centimeters, yeah? So 5 centimeters above the thing should be how far my man moved, you get? So pigment A, yeah, is up there somewhere, yeah? That should be 5 centimeter from the origin, yeah? You get? And then that's over solvent, yeah? So to find the solvent distance here, you need to do 5 divided by 0.95. Yeah, that's gonna get you 5.26 centimeter, yeah? So that means that basically the solvent is 5.26 centimeters, so a little bit above pigment A, yeah? And now you need to find the RF for my man. So for the second thing, yeah? So you know what the solvent is? The solvent is 5.26, yeah? And you know what, and to find the how much pigment C traveled up the thing, you need to again measure it with the ruler, yeah? And you're gonna get something like 3.1, yeah, or 3.05. And then if you do 3.05 divided by 5.26, you're going to get 0.58, yeah? Which is the final RF value, and that's the answer. Alright, cool. Alright, F. The pigments in leaves are different colors. Suggest and explain the advantage of having different colored pigments in leaves, yeah? Alright, calm. So, the advantage of having different colored pigments inside the leaves here is means that it's gonna have more of the range of wavelengths of the light for photosynthesis here. So, try that so they can absorb more wavelengths here of light for photosynthesis. So obviously, yeah, when you got bare colors, yeah, so yellow absorbs one thing, yeah, and reflects the rest. It reflects everything and reflects yellow, yeah, and absorbs everything else, yeah. Same with red, yeah. Red absorbs everything else and reflects red, yeah. So if you got multiple of these, yeah, you're gonna have all the spectrum covered. You get me? So it's gonna be an advantage, yeah. Calm. Question 2A, yeah. So heat stress decreases the light dependent reaction of photosynthesis here. Explain why this leads to a decrease in the light independent reaction. Yeah. So if there's a decrease in the light dependent, yeah, that means that less ATP and NADPH are produced here. So less ATP and NADPH here. So obviously that's what's producing light dependent reaction that needs to be used in the light independent. Yeah. So ATP and NADPH here. So that's all you need to write actually is what is 2 marks. So 1 mark for ATP, 1 mark for mentioning NADPH here. So that's why. It's less ATP and NADPH produced here, so therefore that's why it leads to a decrease in light independent. Yeah. Alright, cool. Question B. Another effect of heat stress is a decrease in the activity of the enzyme rubisco. Yeah. A decrease in the activity of an enzyme means that the rate of the reaction is it catalyzes becomes slower. Yeah. A decrease in the activity of the enzyme rubisco would limit the rate of photosynthesis. Yeah. Explain why. So obviously, yeah, if there's a decrease in the activity of rubisco, yeah, rubisco what it does here is it reacts carbon dioxide with RUBP, yeah. So the CO2 comes in the Calvin cycle, yeah, and then it reacts with RUBP, yeah. So if there's a decrease in rubisco activity, less carbon dioxide, yeah, reacts with RUBP, yeah. So less GP is formed here. So from GP is formed here, and so now less GP is formed. That's all you need to state. That's the 2 marks here, because that means that obviously it's going to decrease the rate of photosynthesis here, since less GP is formed, which means that less sugar can be formed ultimately. Yeah. Come on. Let's see. Where precisely is the rubisco found in the cell? Obviously the stroma. Yeah. Come on. So the scientists concluded that the heat stress reduces the activity of rubisco in plant leaves by affecting rubisco activates. Yeah. Use all the information to evaluate their conclusion. Yeah. So when it says evaluate, yeah, look at the maximum marks, 4 marks here. So what you want to do is you want to mention 4 different things here. Now for evaluate here, what it means is it means mention the pros and the cons here. You need to talk about the reasons why it might support the hypothesis that my man made here and reasons why it might not support the hypothesis. You get me? You need to talk about both ways here. So first thing, yeah, whenever you see these evaluate questions here, I'm gonna tell you how to pattern it. Yeah, listen. Whenever you got these evaluate questions here, it's bait. Yeah, what you need to do first is read the graph here. If it's increasing with the other ones increasing, state that. You get me? State both graphs here. That's 2 automatic marks here. So the first thing that I can see is that if you look on the paper, yeah, in figure 1, yeah, as temperature increases, rubisco activity also increases. So you write that down. So rubisco activity increases here with temperature increase here. That's 1 mark right there, just by reading figure 1, yeah. Then you read figure 2. So as you see, the rubisco activates activity, yeah, decreases at high temperatures. Look at 60 degrees, all of that is getting deleted, fam. So write that down, right? Rubisco activates activity decreases, yeah, high temperatures. Come on. Now, the other marks that you get for the evaluate questions here, bait, bro, bait. Talk about something, yeah, that's not consistent. Talk about how it's a small sample size, yeah. Talk about how one thing only has been tested, yeah. So in this situation, say, yeah, is that if you look at the experiment, yeah, only cotton was used, yeah. So this was used to isolate enzymes in cotton, yeah. So you can say that the results are only for the isolated enzymes, yeah, or you can say that the results are only for cotton, yeah. So you can write that down. So results are only for cotton, yeah. So these are two positive things to enforce it, yeah. Now you talk about two negative things, yeah. So that's only for cotton, yeah, which means that it could be, you know what I'm saying, work differently in other kind of plants and shit, yeah. And the second thing you want to say is that there's no statistical test done, yeah. Listen, bro, that's a easy mark, bro. That's every single evaluate question, yeah. These two are 3 marks, bro. You get me? Evaluate ones are easy, bro, yeah. No statistical test done always gets your mark, bro. Alright, calm. Question 3. Question 3A here. Explain why the scientists measured the rate of production of oxygen in this investigation, yeah? So why did they measure oxygen production, yeah, not anything else, yeah? Obviously, oxygen is produced here in the light-dependent reaction, yeah? Calm. So obviously, oxygen is produced in a light-dependent reaction. So therefore, if you want to measure the light-dependent reaction, yeah, you're gonna need to measure oxygen production, yeah? What you can do is you can even slap a little equation in there, yeah, just to like reinforce your point, yeah, just to show them that you know what you're talking about. You know what I'm saying? Let them know that they can back off, you know what I'm saying? So slapping the little equation here to prove that it's formed in the light dependent here. And you get 2H2 is equal to 4H plus here, plus electron minus to balance the charge here, and then 2O2 as you had two oxygens there, yeah. Alright, calm. Then what you want to say is that, and so, faster oxygen production here means faster light dependent reaction. Yeah, naturally, yeah. The reason you're trying to see how fast a light dependent reaction is, yeah, we're going to measure is the oxygen production, see how fast oxygen production is, yeah. The faster the oxygen production, yeah, means the faster the light dependent reaction, obviously, because that's what's making the oxygen, yeah. Alright, calm. Question B. So in question B, yeah, you want to look at your graph here, and you got a bit of a situation here. So if you look at your graph here, you want to look at the one for low and high light intensity of mutant plants here. So if you read it off the graph here, the mutant plant growing in high light intensity is about 200 here. In the low light intensity is 60. So you want to do 200 minus 60 here. That's going to be 140 here. And now, in the graph it says per hour, yeah? But in the question it says that he's going to measure it every 15 minutes or some shit like that. So therefore, if he's going to measure it every 15 minutes here, so you see it says in each trial the scientist collected oxygen for 15 minutes here, it's hiding above the B, yeah? So what you want to do now is, because that's 60 hours here, 60 minutes is an hour, yeah? And 15 minutes here, think about that. There's 15 minutes in an hour, yeah? 4 times 15 is 60, you get it? So therefore, what you want to do is you want to divide 140 by 4, yeah? To get how many per 15 minutes, you get me? So 140 divided by 4 is going to be 35. So that's 35 micrometers or whatever, yeah? Alright, calm. Micromole per oxygen or whatever the fuck. Alright, cool, 35, yeah? Alright, calm. See, the scientists suggested that the mutant plants producing more chlorophyll B would grow faster than the normal plants in all light intensities. Yeah, explain how how the data supports the suggestion. Yeah, so what you're trying to say is that the data is saying that. Explain using biology, yeah, why that's the case. Why that regardless of the mutation or not, yeah. Why in all light intensities, yeah, the mutation is gonna grow faster, yeah, you understand? So what I would say is that in all light intensities, yeah, chloroplast, yeah, from the mutant plants, yeah, have faster ATP and NADPH production, yeah. So that's in a light dependent reaction, yeah. They got, as you can see, obviously more oxygen produced, yeah. So therefore they're gonna have the mutant plants are gonna have more NADPH and ATP production, yeah. So hence faster light dependent reaction, yeah. She right light independent, yeah, reaction, and so can provide produce, yeah, more sugar for respiration and hence more energy for growth, yeah. So basically, yeah, The reason I said light independent, yeah, not dependent, is because you basically already said that light dependent reaction is going fast in the first statement, yeah. So therefore, you don't need to like state that again. So I said that ATP and NADPH are gonna be made faster, yeah. That's light dependent, yeah. So hence, because they're made faster, there's gonna be a faster light independent reaction as well that follows it, yeah. And so therefore, it's gonna produce more sugar, yeah, for respiration and hence more energy for growth. You get it? Alright, calm. Question 4A, yeah. So explain how two features of the calorimeter, yeah, shown in the diagram would enable a valid measurement of the total heat released, yeah. So first of all, the stirrer, yeah, distributes the heat, yeah. When it distributes the heat, what's that doing? That's basically gonna move it so that it's not like, you get me. If it was in one area concentrated and you put the thermometer in that one area, yeah, it's gonna be overly high, you get me? So that's why the stirrer needs to distribute the heat, yeah, so that it's even throughout the experiment, yeah. And then also, yeah, another feature of the experiment you can talk about is the water, yeah. The water has a high specific heat capacity, yeah. Why use water, yeah? Why not use some other shit, yeah? You're using water because it has a high specific heat capacity, yeah. So obviously that means that it can retain a lot of heat, yeah, without starting to, you get me, without increasing temperature, yeah. Acts as a buffer, yeah, which is what you're needed here, yeah. Alright, calm. So you can put bare heat into the situation without it increasing much. Alright, cool. Question B, yeah. So normally, yeah, more time, yeah, you'd have to convert the centimeter cube yeah into meter cube, yeah. But in this case, yeah, it says that if you look closely, yeah, it says that 4.18 joules of energy are needed to raise the temperature of 1 centimeter cube of water by 1 degree. So in this case, yeah, they're using a specific heat capacity, yeah, that's measured in terms of centimeter cube instead of meter cube, yeah. So that's why you keep it as 100 centimeter cube, yeah. So this is easy, yeah. You just use your energy equation, yeah, which is Q equals MC delta theta. So obviously Q is energy, yeah. So Q equals, yeah, 100, which is the mass of water, yeah. 100 centimeter cube, obviously centimeter cube and mass is the same shit, yeah. Alright, cool. And then 4.18 is the C, yeah. And then delta theta tells you is 15.7 degrees, yeah. So now you times that together, you get 6562, yeah. Point 0.6 joules, yeah. Alright, calm. Now obviously divide that by 1,000 to get kilojoules, you'll get 6.5626, yeah, kg, yeah. And now what you want to do, yeah, is obviously you want to divide that by 2 because you've got 2 grams here. And if you look at the answer in the final thing, it says a kilojoules per gram, yeah. So that's just enthalpy change basically kind of thing. But instead of moles, it's doing it per mass, yeah. So therefore, just divide it by the mass of the substance, which is 2 grams. So that whole thing divided by 2, you're gonna get 3.28 kg, yeah, per gram, yeah. I can't see, yeah. In natural ecosystems, most of the light falling onto producers is not used in photosynthesis. Explain why. Yes, obviously light gets reflected. Yeah, so the reason why not a lot of light is used in photosynthesis. Yeah, is because some of that light is obviously going to be reflected. Yeah, so that's obviously the reason why. Yeah, so you need to think about them things. Yeah, and second of all, is that it's pretty self-explanatory. Yeah, but obviously light is of the wrong wavelength. Yeah, so sometimes the light can be of a wrong wavelength, yeah, since there's bare different wavelengths moving around in the atmosphere, yeah, and so therefore it's not going to be absorbed by the pigment, yeah, because that pigment is not the correct color, or it's actually the light is not the correct wavelength, yeah, for that color. You get me? So for example, if it's green light, it's going to get reflected off the green leaf. You get me? Alright, cool. Question D, yeah. The name two products of the light-dependent reaction that are required. Obviously, we should know this by now. Yeah, ATP, NADPH. Yeah. Hmm. Question E, yeah. Chlorella, yeah, and all them man have a culture of 20,000, yeah, 2,000 man, which was set up in a fermenter, yeah, and they divided every 90 minutes, yeah. You can come. So basically, yeah, times the hours, yeah, by minutes, yeah. So 24 times 60, yeah, to get 1440 minutes, yeah. Cool. Now what you wanna do, yeah, is obviously move my man with the thing, yeah. So you get that many minutes, yeah. Now think about it, yeah, the number of divisions here is going to be that many minutes here divided by how many minutes they divide, which is 90 minutes. It divides every 90 minutes here. So you want to do 1440 yeah, divided by 90 yeah. That's going to get you the amount of times it divided here, which is 16 yeah. Now it's divide when it divides it means it splits up by two. So deeper, yeah. If there was five initial people, yeah, and they divided twice, yeah, it would be five times two. If they divided two times, it would be five times two to the power of two. You get that? Two times two. Then two times two times two. You see that? So what I can say is that initially, yeah, so number of cells here is equal to the initial amount, which is 20,000 here, 2,000 here, times by two to the power of the amount of divisions here, which is 16. So 2 to the power of 16 here. And that's going to get you 1.31 times 10 to the power of 8 cells here. And so that's the final answer. Alright, come on. F here. There is no F. Fam, I fall into that trap every single time. Question 5 here. Question 5A. Calculate the percentage decrease here in the yield caused by the use of atrazine here. Alright, cool. So percentage decrease here. So basically, if you go look at the graph above here, and you go look at sample G, here, sample G here, look at the top, read the top bar here, and it will be 11.7 here, and then read the lighter bar here, and it will be 10.8 here. So what you want to do is you want to do the original here, which is 10.8, because you're finding percentage decrease here. So how much decrease here? So 10.8 divided minus 11.7 here, divided by 11.7, which is going to be the original, yeah? So the original decrease to 10.8. Calm. Then times that by 100, yeah? And then that gives you the final thing. So it'll give you negative 7.7%, but it's just 7.7%, yeah? Alright, calm. B, yeah? So if you don't understand that, what I did is I just took it decreased from 11.7 to 10.8. So you took the original, which is 11.7, take that away from 10.8, yeah? And then divide it by the original, which is the 11.7, you get me? So there you go. It doesn't have to be negative. It doesn't really matter how you do it. Alright, cool. Atrazine, yeah, binds to question B, yeah. So the teacher studying these data with her students told the class that no definite conclusions could be drawn, yeah. So it's just why the teacher said that. So inside that little bar chart, yeah, there's no error bars, yeah. And so error bars are basically, so right, there's no error bars here. The reason you use error bars here is because it's gonna tell you how many standard deviations there are, yeah. So basically draw like error bars above and below, yeah. And if they overlap, yeah, like that, and then this one overlaps with that one, that means that there's no significant difference between them, yeah. So that's why you need to write. So there's no error bars here, which I needed to show if there's overlap, which means it is not significant, yeah. Come. See, yeah. Atrazine binds to proteins in the electron transfer chain, yeah, in the chloroplast here of wheat, reducing the transfer of electrons down the chain. Explain how this reduces the rate of photosynthesis here. So obviously atrazine is going to bind to proteins here. So it literally tells you that, yeah. So therefore, what's the result of that? Obviously electrons can't flow down the transport chain, yeah. Hence fewer protons here are pumped here because obviously you need the electrons to flow through for the protons to be pumped here. So obviously fewer protons are pumped here since fewer electrons are moving down the chain here across the thylakoid membrane here. So there's a reduced proton gradient here because obviously that's what it establishes here. When you get the protons across the thylakoid membrane here, it's going to make a proton gradient here. So obviously there's no proton gradient now here, and hence less ATP is going to be produced here. As NADP can't attach to the H plus now, yeah. So therefore, you can say both ATP yeah, and NADPH are needed here for the dependent reaction. My bad, my bad. Light independent, obviously, yeah. They're obviously made in light dependent, yeah. Cool. So hence, light independent reaction slows down, yeah. That's all you need to write. Alright, calm. So that's perfect explanation, yeah, as why photosynthesis slows down, yeah. Alright, cool. Last question, question D, yeah. When treated with atrazine, yeah, weeds have been shown to give off small amounts of heat, yeah. So just an explanation for this observation, yeah. So obviously the reason why it's going to be given that small amount of heat, yeah, is because the electrons can't flow down the transport chain, yeah, but they're still going to be absorbing light, yeah. So the excited electrons that absorb the light, they're going to be obviously holding heat energy, yeah. And so the energy is just going to be absorbed by the chlorophyll and released into the surroundings instead of being moved down and used to make ATP and shit, yeah. So what I can say is that excited electrons, yeah, cannot be increased, cannot be transferred, yeah. So the energy, yeah, is absorbed by the chlorophyll and released as heat. Calm, yes, that's the reason, yeah. Calm. That's the end of this whole sheet, yeah. Yo, calm. So this is the sheet two, yeah, in the topic five, yeah, respiration. Calm. Question one A, yeah. Calm. So it says here, describe the process of glycolysis here. So in glycolysis here, glucose is phosphorylated using ATP, yeah. So one ATP molecule is used there, yeah, to phosphorylate glucose here. Alright, cool. Then that forms here TP, yeah, triose phosphate, yeah. So each triose phosphate molecule is oxidized to pyruvate, yeah. Calm. So that's the second step, yeah. Removing oxygen, hydrogen, yeah. So obviously when you oxidize here, you remove hydrogen, yeah. So it removes hydrogen, yeah, atoms, yeah, and they are transferred, yeah, to NADH, to NAD, sorry, yeah, forming NADH, yeah. So obviously, bro, you're oxidizing the pyruvate, yeah. So sorry, oxidizing the triphosphate, yeah, to pyruvate, yeah. That's gonna remove hydrogen and transfer that to NAD, yeah. NAD is then gonna attach to the hydrogen, yeah, and make NADH. So basically, in total, yeah, as well, four ATP molecules are getting produced, and two ATP yeah, are being used. So that's the whole process of glycolysis here. Now, listen, fam. Obviously, in the end, you just gotta say that net ATP, yeah. You don't have to say four are produced and two are used. You can say that there's net ATP gain, yeah. But you can just state it like that. Alright, calm. Question B, yeah. Melanate inhibits a reaction in the Krebs cycle. Explain why melanate would decrease the uptake of oxygen in respiring cells, yeah. Calm. So, it inhibits a reaction in the Krebs cycle, yeah. So obviously that reaction that's inhibiting the Krebs cycle here is that no FADH here and no NADH are gonna get produced here. And NADH and FADH2 here are needed here to supply electrons to the hydrogen to the electron transport chain here. And so hence less oxygen is used as the final electron acceptor here. So uptake of oxygen decreases. Alright, cool. So let's break this down here. So what I'm trying to say here is that no FADH2 and no NADH are produced here because of the reaction not happening here. And those are needed to supply electrons into the electron transport chain. And so if that can't happen here, obviously oxygen is the final acceptor here that accepts the electrons at the end. So if there's no electrons adding to the electron transport chain here, then they can't be accepted here, which means there's no use for oxygen here. So the oxygen uptake decreases here. You get it? Alright, cool. Question 2A here. In muscles, yeah, pyruvate is converted into lactate during prolonged exercise. Explain why converting pyruvate into lactate allows the continued production of ATP by anaerobic respiration, yeah? So, the whole point, yeah, when you move pyruvate back into lactate, yeah, the reason why is because it's gonna let NAD get regenerated, yeah? So normally NAD wouldn't be regenerated from NADH, yeah? But you need it to move back there to get NAD regeneration, yeah? So NAD is regenerated. So that means that the process can happen again, yeah? So glycolysis, yeah, can continue, you get me? So glycolysis can continue again, yeah, and you can make ATP again, you get me? So that's the reason, yeah? Alright, question 3, yeah, you got some long diagram. Question 3A, yeah, you've got some long diagram, but the question says just one reason why it's important that the student left the apparatus for 1 hour after the yeast culture at a constant temperature, yeah? Obviously, so that the oxygen inside the experiment can actually be used, yeah? So obviously you need time for the actual yeast to, do anaerobic, aerobic respiration, whatever, fam. And so the oxygen is used. Calm. B. During her investigation, the colored liquid moved to the right. Explain why it moved to the right, yeah? So obviously anaerobic respiration produces carbon dioxide, yeah? Which obviously leads to increase in the volume of gas here inside that beaker here or whatever, inside that tube here, which is gonna obviously push it to the right hand side. You get it? Alright, cool. A student found that the colored liquid here moved 1.5 cms in 24 hours here. The diameter of the lumen of the capillary was 1 millimeter. The volume of the capillary lining is given by pi r squared L, where pi is 3.14 and L is the length here. Calculate the volume of the gas produced here. Calm. So L is 1.5 centimeter, yeah? Just write that down quickly, yeah? So see, L is 1.5 centimeter, yeah? Calm, that's the length here. The diameter, yeah, D, is 1 millimeter. That's going to be equal to about 0.1 centimeters here, according to my calculations here. And so what I can say is that to find the radius, you need to do half of that, yeah? So 0.1 divided by 2, which is going to be 0.05 cm, yeah? Alright, save. Now, V, yeah, according to my man, volume is equal to pi r squared L. So that's equal to pi r, r is 0.05 squared, yeah? And L is 1.5 centimeter, yeah? Use the pi value of 3.14, yeah, and you'll get 0.00, no, 0.11775 cm cube. Alright, safe. Per hour, yeah? There's 24 hours in the experiment, yeah? So that 1.5 was in 24 hours, yeah? So per hour, you need to divide that whole thing by 24, yeah? So divide that by 24, you're going to get 4.9 times 10 to the power minus 4 cm cubed, yeah? Per hour, yeah? Calm, that's the final answer. Alright, calm, safe D. So it gives you a log kind of scale, yeah? And it says, explain why a log scale is recorded, yeah? So the reason it's a log scale is because the number of cells is going to increase exponentially, yeah? So deep it, how many cells are getting made here? There's bears here, and obviously you can't have that. That's going to be like 3 times 10 to the power 50, whatever. And it's going to be big numbers here, so you need to basically bring that down into like 1 point something, whatever. So you're going to need to log it, yeah? Alright, come, question E. Many yeast cells die during the death phase, so just one reason, yeah? Obviously competition for glucose, innit? So competition for food, yeah, there's not enough glucose around here, so obviously they're going to die out. Alright, come. F. So you got some situation, yeah? I need to pattern it. So what I would say, yeah, is that it says xt is equal to x0, yeah? E to the power of rt, yeah? So find rt, yeah? So rt is just 0.5, yeah? Which is the growth rate, r, yeah? Times t. T is in hours, yeah? It's 10 hours, yeah? So that's 5. So rt is just 5, yeah? Come. So xt, yeah, is 2000, yeah? E to the power of 5. Put down your calculator, yeah? You'll get 296,826, yeah? Come. If it doesn't let you do that, yeah, put e to the power 5 and then times it by 2000. Do a and s times 2000, yeah. Alright, question 4. AMP activated protein kinase, yeah, AMPK, yeah, is an enzyme that regulates the number of cell processes. Exercise leads to activation of AMPK. So basically, when you got AMPK, yeah, it leads to inhibition, yeah, of melanocortin-A, which leads to inhibition of CT1, CPT1 transport, yeah, into the mitochondria, yeah. So CPT1 is a channel protein that transports fatty acid into mitochondria, yeah. Using the diagram above, explain the benefit of AMPK during exercise, yeah. So the benefit of AMPK, yeah, is that you're gonna get less CoA, yeah. So it produces less melanocortin coenzyme A, yeah? Now, why is that a good thing, yeah? That's a good thing because melanocoenzyme A leads to an inhibition of CT1 moving to the mitochondria, yeah? So that means that if there's less of melanonal, yeah, and melanonal is moving the shit into there, not allowing it to go into there, there's gonna be more shit moving into there, you get it? So more fatty acid, yeah, are transported, yeah, into the mitochondria. You understand? And hence respiration. Hence these ATP, yeah. Sorry, these fatty acids can be used as respiration, yeah, to make ATP. Yeah, you get it. So less melanonal, yeah, obviously means that there's gonna be less inhibition, yeah, which means that more is gonna be transferred to the mitochondria, so more respiration and producing more ATP, yeah. Come on, question 5A. The first 10 minutes here, the tap attached to tube A was left open and the syringe from tube B was removed here. So just three reasons why the apparatus was left for 10 minutes. So it's easy, yeah. You just write to allow the apparatus here to equilibrate. So everything needs to become level again, yeah? And then also to allow the respiration rate of the seeds here to stabilize. So to allow them to respire at a constant rate, yeah? And then also, yeah, to allow the pressure to equalate, yeah? So to make the atmospheric pressure outside equal to the inside, yeah? Alright, come on. B. So just to explain why the chosen temperature was 20 degrees, yeah, for this experiment. So why 20 degrees is obviously the one that you want to use here is definitely because it's just fucking going to be the optimum temperature in it. So write that down. So optimum temperature, obviously, yeah, for growth of seeds, yeah. And then also it's going to be optimum temperature, yeah, because you're investigating respiration and enzymes and shit like that, yeah. So optimum temperature for the enzymes involved in respiration, yeah. So that's calm, bro. That's easy. Alright, cool. And question C, yeah. Alright, part C, yeah. During the experiment, the colored liquid in the tube being moved towards tube B, yeah? Explain what caused this, yeah? So obviously oxygen is going to be used up by seeds, yeah? And then the CO2, yeah, is going to be released in it. So obviously oxygen, yeah, is used up by the seeds. And the carbon dioxide, yeah, is released and absorbed, yeah, into the solvent, yeah, the solution, yeah, which is KOH in the situation. So hence the pressure, yeah, in B decreases, yeah, you get it. So it's going to be absorbed into there, yeah, so the pressure inside B is going to decrease, yeah. Alright, calm. Question D, yeah. The mass of C's was 1.6 grams. Use the information in the table above to calculate the rate of oxygen consumption in centimeter per cube per whatever, yeah, at calm. So look above here, and you'll see that you want to do it from 1.6 here. So you can take it from obviously the most like accurate data, yeah. So take it from 0 to 10. So for 0, yeah, the plot is 0.84, yeah, on the sheet. And for 10 is 0.58. So do 0.84, yeah, minus 0.58 divided, yeah, by the that's a centimeter cube, yeah. You want to obviously divide that by the gram per hour, innit? So divide that by the gram per hour. There's grams is 1.6 here, and then divide it by hour. That's for 10 minutes, bro. That's from 0 to 10 is 10 minutes here. So 10 divided by 60 is gonna give you how many hours in 10 minutes here. So 10 divided by 60. So what you can do in other times is that divided by that, yeah. That's the amount of cm cubed, yeah, made here. Divide that by 1.6 amount of grams here, and then divide that again by 10 over 60, yeah. Or you can just put 10 over 60 times 1.6 on the bottom, and it does the same thing, yeah. And put that in your calculator, you'll get 0.975 cm cubed per g per hour, yeah. Calm. Question 6, yeah. Describe the advantage of the Bohr effect during intense exercise, yeah. Calm. So the Bohr effect here, what it does here, is it's going to increase the unloading of oxygen, yeah. So it's going to make oxygen unload faster, yeah. So increases here, unloading, yeah, of oxygen. Calm. For aerobic, yeah, respiration in the muscles, yeah. Reason you need to state for aerobic, yeah, is because anaerobic can still occur, yeah, without oxygen in it. So you need to say aerobic, yeah, specifically, yeah. So you can say that as for obviously times where oxygen is not available, yeah. Like when you got bare CO2, yeah. Alright, cool. You want to do question B now, yeah? So question B. Question B, yeah, is easy, yeah. It says that VT can be identified, yeah, as the first point where there's an increase in PO2, but there's not an equivalent increase in CO2, yeah. So if you look at where that is, yeah, where is there a strong increase here in oxygen here on the graph here, but not with carbon dioxide here? So the top line needs to be going up and the bottom line needs to be just be doing nothing here. That's obviously at time equals 10 minutes here. If you look at the bottom where it says t equals 10 here and you look up here, that at that point you see how the top line is going up and the other ones are not doing anything here. So that's what you want to do here. So at that point you want to read it off here. Each box here is about a zero point 0.4 increase. You get me? Each box is zero point 0.4 basically going up here. Now be careful because the left hand side starts at 8 here and the right hand side starts at 4. So remember to get the amount of oxygen here, you need to look at the left hand side of the graph here. And to look at the amount of CO2, the scale was the right hand side of the graph here. So if you look at that here, the pressure of oxygen here, if you read it from the left hand side, is going to be 10.8 at 10 minutes here. And the pressure of CO2 yeah, at 10 minutes, if you read it from the right hand side, is 6.4 yeah. So now obviously divide by the smallest one there. So divide by 6.4, which is 1. And this divide by 6.4 is 1.6875. So that's the final answer here. Alright, cool. Question C here. So question C is talking about um so just one physiological change here that would cause this result. So you're saying that the graph shows that the PCO2 is air did not show a large increase during the exercise here. So you're making more. What he's saying is that during exercise here, you made more CO2, but there's not an increase in the pressure in CO2 built out yet. The reason for that is because your breathing rate is going to be increasing, but you're not going to suddenly be breathing out more carbon dioxide here just because you're doing exercise. You're going to be, breathing out the same amount of CO2, but you're going to increase your breathing rate. So there's more breaths here, so you're releasing more CO2 in total here, but each one's staying the same. You get me? So just write that down. So increase in breathing rate here. However, similar PCO2 for each breath. Come. Question C here. D even, fam. And more than 80% of maximum muscle effort, yeah, ATP, yeah, can only be made for a limited time, yeah. Use the diagram above to suggest one reason why, yeah. So the answer, yeah, is that muscle fibers have a limited amount of phosphocreatine, yeah. So the second box here, so muscle fibers have limited phosphocreatine, yeah. No, the reason for that, yeah, is that's just bait, fam. Obviously, there's a limited amount of muscle and phosphocreatine inside the body, yeah. That's why you take creatine powder and that sort of shit to increase it. You get me? Alright, cool. Yeah. So you got some drug, yeah, and this drug activates AMPK, yeah, and develops slow muscle fibers at rest. Use the diagram above to justify why professional athletes are not allowed to use the drug, yeah. Did not include details of chemoosmotic theory. So what you want to do, yeah, is go look above at AMPK, yeah. It's gonna make more AMPK, yeah. So if there's more AMPK, if you look at the left-hand side of the diagram, yeah, it says that there's more fatty acids in the muscle fibers are oxidized into acetyl coenzyme A, yeah. So more acetyl coenzyme A is made, yeah. So just write that down. So more acetyl coenzyme A, yeah, enters the Krebs cycle, yeah. As you know, the acetyl coenzyme A enters the Krebs cycle at the top, yeah. And so obviously more production, and then obviously binds to whatnot, and then it produces NADH and FADH2, yeah. And hence more ATP, yeah, obviously. And so hence athletes, yeah, build slow muscle fibers without exercise, yeah, because it says that it does it for the slow one, yeah. So what's going on here, bro, is normally you'd have to do exercise for that to happen, yeah, for it to stimulate that and then do more Krebs cycle, yeah, to get more ATP produced, yeah. But you're making ATP without actually doing anything, yeah, just by taking the drug, because it's gonna make more acetyl coenzyme A, yeah. So that's basically why it's unfair advantage, yeah. Because obviously you can just take that, yeah, and have bare ATP, and your body will know what to do in times of like that. You get me? So yeah, calm. And F. EPO, yeah, is another performance enhancing drug. It can increase the percentage of red blood cells in the cell, yeah. A heart attack is caused by a lack of glucose and oxygen being delivered to the cardiac muscle via the coronary arteries, yeah. The overuse of EPO can increase the heart attack. So why do you think, yeah, something would increase the rate of a heart attack here? It's probably because the coronary artery is going to get blocked here. The only way it can block it, it gives you a little bit of a clue. It says that it's going to increase the red blood cell percentage here. So if it's increasing the red blood cell percentage here, it's going to become more viscous. You get me? It's going to have more like red blood cells here inside the water, yeah. You get me? So not enough water, yeah, for the red blood cells. You see what I'm saying? So it's going to make it slower, fam. It's going to thicken the blood. You get me? So EPO, yeah. See, to use a bit of logic here, EPO causes the blood to thicken, yeah, which could slow blood flow, yeah, and hence cause the heart attack. Calm. Next question, yeah. Calm. Question G, yeah. So it's asking why that's unfair, yeah. So the normal hematocrit, yeah, for human males is 47%. For professional cyclists, the hematocrit, yeah, is allowed is 50%, yeah. So basically, yeah, if you're taking, bruv, this situation, you're gonna get up to 50% if you're taking EPO, yeah. If you're taking EPO, that's 50%, and it can also be naturally above 5% as well. So you can get up to 55% if you're taking the drug, yeah. Which means that you're 8% more than 47, so you're 8% more than the other man, yeah. So that's why obviously it's not fair in it, because some cyclists here will have 8% more than others, yeah. And the other reason as well why it's bad, yeah, is that you can die from it, innit, fam. So obviously health risks, yeah. Alright, calm. Alright, cool. 7A, yeah. So we've got a table here and it says find the rate of carbon dioxide per gram, yeah. So you need to do rate of carbon dioxide divided by the mean mass here. So do that for each one, yeah. So 0.12. You're gonna get 2.56. Next one, yeah, 0.33 divided by 0.046, yeah. that, yeah, and then 11.67, yeah. Then what you want to do, yeah, you want to put temperature on the y-axis, yeah. No, rate on the y-axis, temperature on the x-axis, yeah. And you want to draw this graph here, so make sure you got these increments here, and then increments up here, label it, yeah. And then it's going to be a straight line graph here, so just draw the points and connect it by a straight line, yeah. And that's it. Alright, calm. Question B, yeah. So question B, yeah, it says calculate the change in rate per hour of opening or closing the spiracles, yeah. So what you need to do, fam, is basically this one's like looks weird in it, but it's actually easy. You go up to the top diagram, 10 degrees here. How many do you see? Like on the bottom, you see how on the bottom of all three graphs here, all the way at the bottom here, it says 60. Just look at the 60 where it says like 60 and go up from there and count how many. So in the first graph here, there's three and then 60 is like there. So that means that there's three basically, fam. In the first one, yeah, in 20 degrees, in 10 degrees, yeah. Then look at 20 degrees, yeah. There's like 11 things, yeah. But then the other one cuts it off at like 10, fam. You get me? But in the mark scheme, it says it as 11, fam. So what we want to do is 11 minus 3, yeah, which is going to be 8, fam. So therefore, there's 8 per hour. Obviously, you're doing it per hour. That's why you looked at the 60 mark here. Alright, cool. Let's see, yeah. Fine, explain how you could determine, yeah, the total amount of carbon dioxide secreted at 30 degrees, yeah. So just calculate the area under the surface here, under the graph here. So reason for that, yeah, is obviously because like if you times the bottom by the top here in minutes here, it's gonna cancel the minutes. You're just gonna get the micro dm cube here, which is the amount of carbon dioxide in it. Alright, come on. Question D. So just an explanation for the effect of temperature on the rate of carbon dioxide release here. So obviously as temperature is gonna increase here, enzyme activity here and metabolic rate here also increase. Hence here, this leads here to a higher rate of respiration, producing more CO2, yeah. And so to remove the extra CO2, yeah, the spiracles, yeah, are more frequently staying open, yeah. Understand that, fam. So to remove the excess CO2, yeah, the spiracles are gonna open more frequently, yeah, and that's why it's gonna increase carbon dioxide release, yeah. Alright, come on, that's it. Say if there's a third sheet inside topic 5 here, so energy and ecosystems here. Alright, question 1A. So it says here, use your knowledge of MPP here to explain why freshwater materials have a high CUE. Yes, obviously these freshwater ones here, they're not gonna be doing that much respiration in it. So obviously that's why less respiration, yeah. So therefore less energy used up in it. So therefore more growth here. Doesn't take much respiration to get my man functioning. You get me? So it's not gonna use that much energy. You see? Alright, calm. Alright, cool. Question B here. So it says here, freshwater marshes soils are normally waterlogged here. Use your knowledge of the nitrogen cycle to suggest why these solids contain relatively high concentrations of ammonium compounds here and low concentration of nitrite and nitrate here. So obviously there's less oxygen available here for nitrifying bacteria and hence less nitrification here. So there's less conversion of ammonium ions into nitrite ions here. And then again into nitrate ions here because they're going to add another oxygen here. Yeah. So C is saying state the assumption that the student has made, yeah, and suggest why he's done that, yeah. So obviously my man substituted, yeah, height for biomass, yeah. So obviously he can't do that, yeah. So what he's done there, yeah, he's assumed that height is proportional to biomass, innit? Obviously the roots here are also going to be under the ground, yeah. So therefore measuring the height is not going to account for that. So therefore that's going to be a bit of biomass here that was let out, you get me? So that's why it's invalid here. Alright, cool. Yeah. So you need to find W1, yeah. So it's saying that the difference was 268, yeah. And it said that and the end it was 387, yeah. So to find the initial, you're gonna need to do 387, yeah. So W1 is gonna be 387, yeah, minus 268. If you pattern that, yeah, you're gonna get 119, yeah, which is therefore W1, yeah. Then you need to find W2. W2 gives it to you, yes, 387, yeah, it's the final one, yeah. Come. And R, yeah, is also given to you, yeah, is 0.097. So now you just need to pattern the full thing, yeah. So obviously you move my man here, so R is equal to LNW2, yeah, minus LNW1, yeah. All of that over T, yeah. So obviously what I can do here is I can say that move my man up there and then divide by R, yeah. So T is equal to LNW2, yeah, minus LNW1 over R, yeah. Alright, calm. Then obviously put inside the values, yeah. So ln 382, 87 fam. Take away ln, yeah, of 119. All of that divided by 0.09. And now you're gonna get left with like a small little answer, yeah. That answer is being at 12.4114, yeah. So 12.14 days is equal to roughly 12 days, yeah, according to my estimations, yeah. So there you go, fam. Alright, calm. Question 2, yeah. Question 2A, yeah. So it says here that you need to compare, yeah, using potassium nitrate compared to ammonium sulfate, yeah. So there'll be like a graph here. And if you read the triangle one, yeah, the triangle one cuts 37.5. So read 37.5 on the bottom, yeah. And then that cuts the triangle, yeah, at like around 4.9. And it cuts the square one, yeah, at around 3.5. So you do 4.9 minus 3.5, yeah, is going to be 1.4, you get me? So the answer is 1.4. Can't be. Says using all the information, evaluate the effect on the plant growth here of adding the different fertilizers to the soil. Yeah, so you need to basically, when it says evaluate here, how many marks is it? Yes, 5 marks here. So you need to basically say the trends inside the graph here. So what increases here, and what decreases and what not. And then you normally say no statistical test, that kind of thing. Yeah. Alright, calm. So basically, yeah, write down potassium nitrate. Yeah. So if you look at the graph here, the potassium nitrate is the most effective fertilizer clearly since it led to the most growth here. The chicken manure, yeah, that's the least effective in it. So as you can see on the graph here, it's obviously the least effective one because it led to the lowest amount of increase in biomass here. You can also state here, all the fertilizers, no matter what, are more effective than control than having nothing, yeah? You can continue to reach it off the graph here. So say that for potassium nitrate, So look at like key points inside the graph here. So another key point is that for potassium nitrate here, it increases up to 30 Gs here, but then there's no further increase. So um, now you can see that ammonia sulfate here. Showed a gradual, yeah, steady increase here in biomass after 30 Gs, yeah. Because if you look at 30 Gs, the one for ammonium sulfate, yeah, has like a little increase after that, yeah. Cool. And then decreases after 45, yeah. Safe. So last thing you can say as well is that this is only done for spinach. So for other plants, it could be different, yeah. Always, that's what you need to do in evaluation, yeah. You need to talk about what could be different, yeah. And the final thing, of course, is that no statistical test, yeah. Calm, that's all of it, pattern fam. Part C, yeah. So the scientists determine the dry mass of spinach first to heat a sample, yes. Just why the scientist should do to ensure it has removed all the water, yeah. So he's gonna reheat, yeah, until the sample remains the same mass, yeah. So he's gonna reheat it, yeah, and he's gonna keep weighing, yeah, until a sample is the same mass, yeah. So reheat and keep weighing, yeah, until it stays the same mass. That means that all the water's been effectively removed, yeah. Alright, calm. Question 3A, yeah. So use the diagram to calculate the percentage of sunlight energy that would be transferred to the feces and the urine of a zebra, yeah. Calm. So if you look at the first percentage, that's 2.1%, yeah, which is 0.021, yeah. Times that by the second percentage, yeah. If we're going from a vegetation to a zebra, that's another 0.127%, yeah. So times that by 0.127, yeah. Calm. Now you got that. Times it again. Gonna go to the feces and urine, yeah, which is 58%. So that's 0.582. That's patterned there. So that's gonna get you like roughly 0.0015552. If you times that by 100%, yeah, that's gonna be 0.15% basically. 155%. Calm. Part B, yeah. So part B says, yeah, it's actually part B and C, yeah. So it's actually part C, yeah. Calm. So this says, yeah, the amount of vegetation is 24525, yeah. And you need to move it from zebra. First, you need to move it from vegetation into zebra, yeah, which was 12.7%. So times that by 0.127, yeah, and that's gonna get you 3114.67, yeah. The reason I times it by that is because I'm dividing by 100, yeah, to get the percentage back into decimal, yeah. So I've got 3114.67, yeah. Now you need to move zebra into new tissue, yeah. So 3114.67 times it by a percentage for new tissue, which is 0.057, yeah. And you're gonna get 177, which is roughly 180, yeah. Alright, calm. Question 4, fam. So just one way in which the increase in uptake of phosphate could increase plant growth here. So obviously that's gonna be producing ATP, yeah. So it's used to produce ATP, that's what's used in plant growth here. Alright, calm. Fam. Alright, B. So just one way in which AMF may benefit from an association with plants here. So AMF is some kind of like fungi, bacteria, whatever, yeah. So that's gonna need obviously require sugars in it from the plant. And so it requires sugars produced by the plant. Obviously, it helps the plant because it increases the root surface area and that kind of thing, yeah? Alright, calm, see. Explain why the increase in shoot biomass here can be taken as a measurement of MPP, yeah? So, shoot biomass here is literally dry mass, you get me? Because it's not respiring, yeah? So it's included without the respiratory like losses here, you get me? So it's literally just a straight dry mass here. And then you can quote that represents here gross production here minus respiratory losses here, because that's what MPP is, yeah? Calm. Question D, yeah? Using the data, turn the graph in part C, evaluate the effect on plant productivity of adding AMF here. Calm. So in the control one, yeah, an increase in phosphate increases plant growth. Mhm. And then for the other shit, yeah, the enterophosphora, yeah, it's gonna do the opposite shit, yeah. So obviously my man's gonna reduce plant growth here when you add more phosphate in it. Now I can also state, yeah, the classic here, you already know what it is, bro. No statistical test done in it. I can say as well that it's only 20 weeks of growth here, so it could change over many, many weeks here. So it's not enough data here. I can't E. So E says using the E button, yeah, because that's obviously what you're going to use to inverse a log here. Determine the root of biomass production per day for the control plot with normal, yeah. So if you look at normal, yeah, it's about 2.4, yeah. So if you look at normal on the left-hand side with no AMF here, it's about 2.4, 2.5, you get me? So what you need to do is write E to the power of 2.4, yeah, because that's how you inverse a log, yeah, is equal to, because this is E to the power of log 2.4, so we'll cancel it, yeah. That's equal to 11.023. And it's per 20 weeks, yeah. 20 weeks in days is 20 times 7, yeah. So you need to do 11.023 divided by the amount of days, yeah, which is 20 times 7. And that's going to get you 0.079, which is roughly, yeah, equates to 0.08. Calm. Question 5, yeah. So the total mass of wood sample is due to loss of water. Water has a density of whatever, yeah. Use the data in the table to calculate the percentage of water freshly out of wood sample, yeah. Sure you're working. So to find the mass of fresh, yeah, I'm gonna call that mass of F. You need to do the density, yeah, of F, fresh, times the volume of fresh air, which is in the table 993, is the density of fresh air, times by the volume of fresh air, which is 1.345. That's going to get you 1335.585 grams here. So you got the amount of G's of fresh air. Now you need to find the mass of dry air. So mass of D here, that's going to be the same thing here. So density times volume here. So density of D times volume of D here. So that's again going to be the density of D is 769 and times that by 1.125 here. That's the volume of D. So you're going to get 865.125. That's the amount of G's of that here. Confirm. So therefore the mass of water here is basically going to be the mass of the thing that's fresh here with all the water minus the mass of it dry. Because that mass would have evaporated here. That's the water itself here. So to find the mass of water always here, do mass of hydrated minus mass of dehydrated here. So 1335.585 minus 865 0.125. That's gonna get 470.46 G's. Alright, calm. Yeah, so it's asking you for the percentage of water, yeah. So obviously divide that by the total mass now. So do 470, yeah, 0.46 divided by the mass of the fresh, yeah, because that's over which one had water, yeah. So 1335.585 times 100, yeah. That's gonna get you the percentage water, yeah. So that's 35.23%. Alright, calm. Next one, yeah. Question B. So saying that ecologist here had some situations here, and it's asking how he knew, like, or how he knew all the water left here. So that's standard procedure by now. I already know what to do here. You record here the mass and the reheat until a constant mass is established here. Let's save. Question C here. So what does the graph show about the relationship between the diameter of the trunk and the trees and their biomass here? So there's obviously a positive correlation here. So that's what you need to say here. Positive correlation here. And also non-linear, yeah. My man's a curve here, so obviously my man's not a straight line, so it's non-linear, you get me? You could also say exponential as well. Calm, D. So we got plantations of trees here are often created to remove carbon dioxide from the atmosphere yeah, to help to balance the carbon dioxide released by burning fossil fuels here. For different species of tree, information is available for the relationship, the percentage, and the mean dry density here, using only the information provided in Part C, such as how the mass of carbon in the wood can be measured here. Alright, calm. So what you're gonna need to do here is obviously measure all the trunk diameters of the trees here. And then calculate the mean diameter here, and hence estimate the mean fresh biomass here. So basically, yeah, there'll be like a table in real life here, and then probably diameter is obviously proportional, yeah, to the biomass of the trees here. So you just look at the table, and whatever diameter you get, yeah, you get the biomass of the tree relating to that. So then use the percentage of water after this to calculate the dried biomass here. So what I mean by that is if the percentage water here is like 30%, then you're gonna find 70% of it, which is the dry one, because 30% will be water, so the rest is dry, so 70% here. And then count the number of trees here and times by the mean dry biomass here. So that's gonna tell you how much dry carbon is in all the trees here, which is gonna tell you the carbon content here. Alright, calm, that's it. There's fourth sheet inside topic 4 here, topic 5 here, photosynthesis here. It's called nutrient cycles here, pattern. Question 1A. So it's talking about these freshwater marshes here, they have one of the highest rates of GPP here. So the reason for that is because obviously there's little respiration here. So there's going to be less energy loss here in respiration here. So therefore more growth here. So it's going to be growing more because there's less respiratory losses here. Alright, calm. Question B, pattern. Use your knowledge of the nitrogen cycle to suggest why these soils contain relatively high concentrations of ammonium compounds and low concentrations of nitrite here and nitrate ions here. So obviously less nitrification here, as there's less oxygen available here, and so less conversion of ammonium ions into nitrite ions here. So NO2 minus here. NO3 2 minus is nitrate here. Come on. C. So it says here, use your knowledge of the nitrogen cycle. Nah, that's part B, yeah. So C is saying state the assumption here that the student has made and suggest why this assumption might not be valid here. So obviously the assumption that he made here is he replaced the um biomass here with height here. So what he's done there is he said that the student has assumed here that height is directly proportional to biomass here, but height does not include the roots here. Because obviously you're going to get the roots here, which are underneath the plant here, so you can't obviously see that by the height, you get me? And that's a little bit of biomass that's been neglected, you feel me? So there you go, yeah. Question D, yeah. So we got Ellen's situation here, you got to find my man, yeah. So what you need to do here is bait, yeah. You need to find W1 first here. W1 is going to be the final, yeah, which is 387, yeah, minus the thing that changed by, which is 268, yeah. That's going to get you 119. And then with the 1 white, you need to find W2, yeah, which is going to be 387, yeah. That's the final amount that it equates to, yeah. And R is going to be 0.097, yeah. It gives us that standard, bro, yeah. Alright, calm. So you got R is equal to ln W1, yeah, minus W2, yeah, minus ln W1. All of that over T, yeah. So obviously going to replace T with R, yeah. So you're going to get T equals ln W2, yeah, minus ln W1, yeah. All of that divided by R, yeah. Alright, safe. Now what you want to do, yeah, is you want to put in the values that you got. So that's going to be T equals, yeah, ln 387, yeah. Take away ln 119. All of that over 0.097, yeah. That's going to get 12.14, yeah, which is basically 12 days. Alright, come. Question 2, yeah. Describe the role of separate biomes in the nitrogen cycle, yeah. So obviously these man, yeah, they're gonna use enzymes here to decompose the proteins here and produce ammonia, yeah. So come. Question B, yeah. So question B says here, you're given a sample of water from 3 different rivers here. Describe how you could obtain a quantitative measurement for the cloudiness here. So that's gonna be using a colorimeter, yeah. Color, yeah, meter, yeah. Alright, come. To measure the absorbance here of light. So basically, yeah, obviously you said this is cloudy and the other thing, that's gonna be subjective, yeah. That's quantitative, yeah. So this one, yeah, it's gonna be quantitative data. The other one is qualitative, yeah. So this one is gonna be like actual number of absorbance of the light, how much light is absorbed through the sample. You get me? Obviously, less light that's absorbed, the more cloudy it is, yeah. Calm. And also, yeah, and make the kilometer, yeah. Make sure, yeah, at zero, yeah. Zero would mean that's set to zero before you do it, yeah. That's gonna standardize the test, yeah. Calm. Question 3A. So the scientists use that micrograms per G, yeah, for the concentration of ammonia. So it's just why this investigation, the scientists use these units, micrograms, yeah. Obviously, bro, there's gonna be like trace amounts of ammonia here in the soil. Yeah, it's not like there's bare ammonia or nothing. Yeah, so that's the reason why you're gonna be using micrograms here. So because so there's very little ammonia inside the soil. Yeah, that's why they're using micrograms. Yeah, and the other thing, G to power -1. Yeah, is to allow for comparison between samples. Yeah, Because obviously each sample is going to have a different amount of grams here. So therefore, if you divide it by the amount of grams, you're going to account for that, yeah? You're going to see per gram here. Can't be. Calm. So B saying 0 and day 2 here from A and B here. So look at day 2. And then 2 boxes from the right here is where you're going to want to look here. That's going to be day 2. Yeah, so 2 boxes from the right here. Then you want to go 11 up here. So it's going to be roughly 11 and 2 here. So for A is going to be 11. For B is going to be 2. So then what you want to do here is obviously take that away. So 11 minus 2 is going to be 4.5. This year. Not 4.5, sorry, 9 here. And then after 11 minus 2 is 9, you're going to divide it by the number of days here, which is 2 days here to get 4.5. This per day as well. You get it. So day to power of minus 1 here. Alright, calm. Question C. The scientists conclude that the soil mixture from experiment showed there was different communities of bacteria in the soils A and B. Here. What evidence from figure 1 supports their conclusions here? So obviously the reason here that it's going to be different is that um B here with pH 4.3 was the fastest here rate of breakdown. Yeah, so if you look at the graph here, it's breaking it down bare fast here. Calm, bro. And then the A plus B here was slower. The A plus B mixture here was slower than A or B here. So A on its ones or B on its ones here. A plus B is slower than both of them here. So hence here, what that means here, the pH 4, so the B one here, the pH 4.3, doesn't work well here at pH 9.6, 6.9 here. So that's what proves that there's going to be two different communities, yeah? Because if you put it on as ones here, they both work well, yeah? When you put it together, they're mixing the pH now, you get me? So it's not going to work at that pH. So the one that was in 4.3 that was doing it bare fast here is going to slow it down to become even slower than both of them, yeah? Alright, calm. D, yeah? So if you look at S in the diagram, yeah? So S went from 10 to the power of 6, yeah? So 10 to the power of 4, yeah? So therefore, find the percentage difference between S here. So S is going to be, yeah? Remember, it's the original year, which is 10 to the power of 6 here, minus the new one here, which is 10 to the power of 4 here. All of that divided by the original year, which is 10.6. 10 to the power of 6 here, times that by 100 here. You're going to get 99%. Do the same thing for T now, yeah. T went from 10.2 to 10.4 increase here. So T is equal to 10.4 here, which is the final year, minus 10.2, divided by the original, which is 10.2 here, times 100 here. It's going to get you 9,990, 9,900% here. So therefore, T is the one that made the most significant difference here because there's a bigger percentage change here. Calm. Question E. This method allowed the scientists to estimate the expression of the AMOA gene in each culture, but not the growth of bacterial production in each culture. Yeah, explain why. So growth of bacterial culture is cell division, yeah, so it's making more cells, and then yeah, you get me. What they did here is they measured the mRNA, yeah. So mRNA is not going to tell you how many cells there are, it's not going to tell you how much it increased, you get me. So you need to write they didn't count the number of bacteria. You'd say mRNA amount here relates to enzyme amount, not cell number. So basically, yeah, just because there's more mRNA here doesn't mean there's more cells here. It could just be that there's more enzymes to break down and could produce the mRNA here. Cool. Also, we don't know how much mRNA each cell has here. So you can't just divide the total mRNA, yeah, produces, yeah, by the number of cells, yeah, because like by the amount one produces, yeah, because each cell is going to be producing like very different amounts of mRNA, yeah. So therefore, it's not gonna having the mRNA doesn't tell you how many cells there is, yeah. Also, yeah, we don't even know, yeah, if this a MOA situation, yeah, is actually linked to cell division, yeah, to cell growth, yeah. Could be linked to some other shit. Yeah. It doesn't necessarily mean that the more you have, yeah, the more that you're growing, yeah, the more cell division, yeah. Alright, cool. F, yeah. The scientists set up the cultures, yeah, in sterile glass bottle, yeah. So just one way to sterilize the bottle, yeah. So what you want to do to sterilize it is you want to obviously move on, wash it in alcohol, yeah, to kill all the bacteria, yeah. Calm. Question 40. So give two examples of biological molecules containing nitrogen that would be removed from when a crop is harvested, yeah. So easy two ones here, DNA, RNA, yeah. Obviously there's bare things that you can say, yeah. Calm. B, yeah. So you need to find out, yeah, the difference in mass of game produced, yeah, per hectare, yeah, in 1970 with 2005. So obviously you want to use the fertilizer response ratio, yeah. So fertilizer response ratio, it says is kg grain produced for each kg fertilizer, yeah. So kg grain produced for kg fertilizer, yeah. So what you want to do is you want to find kg grain, yeah. So you're gonna need to take the ratio and times it by kg fertilizer, yeah. That's gonna get you kg grain, yeah. So what you want to do, yeah, is you want to come here. You need to see for 1970, yeah. So for 1970, you want to read it from the line. So if you read it where the line graph is, yeah, not the bar chart, yeah. Where is 1970? It's gonna be about if you look at the middle where that first X is, yeah, it's gonna be about 14, yeah. So 14 kg fertilizer. So 1970 is 14 kg fertilizer, yeah, of F. And if you look at 2005 on that line graph here, it's gonna be about 75 kg fertilizer. So just look at the left. Remember to use the left graph for the fertilizer, the right graph for the ratio, yeah. Calm, bro. Now the bar chart, yeah, relates to the ratio, yeah. So you want to do 1970 bar chart, yeah. So 1970, if you look at the bar chart, it's gonna be 12.4, yeah. Remember, use the right hand side of the graph to use it, yeah, the y-axis on the right, yeah. So 12.4 kg per whatever. And in 2005, the ratio was wait, let's say it was like 3.6, yeah. So now find out the amount for both of them, yeah. So times it, yeah. So for 1970, do 14 times my man, yeah. 14 times of 12.4, yeah, you're gonna get 173.6. Then do the same shit for 2005. You get 2005 is equal to 75 times 3.6, yeah. You're gonna get 90, no, you're gonna get 270, yeah. Then do 270, yeah. Now you got the mass for both of them, yeah. So you need to minus them away. So 270 minus 173. It says from 2005 in the question, so it gives you a hint that the 2005 one's gonna be bigger, yeah. So that's gonna be 96.4, yeah. That's the final answer. Alright, cool. C. Use the data and the graph above to evaluate the use of artificial on grain producing crops in India. Yeah, so obviously the fertilizer, yeah, is going to become less effective over time as seen. As the time increases, it becomes less effective. Yeah, so the fertilizer, you can see here that there is correlation here in the growth rate. So it is help the fertilizer is helping it grow. Yeah, but the graph here doesn't prove. the changes in yield, yeah, are due to fertilizer. Because it's just showing the fertilizer and showing the yield, yeah, it's not actually showing you like all the other factors that could be affecting it, yeah. So that's what it means by evaluate, yeah. You gotta talk positive and negative, innit? Alright, calm. Question 5, yeah. Explain why the scientists sterilized the surfaces of the seeds, yeah, and grew them in soil that had been heated to that. So the reason they sterilized the surface of the seed is because the surface is gonna have bacteria. The reason they heated the soil is because the soil is gonna have bacteria, yeah. So to kill any bacteria, yeah, in the seeds or in the soil, yeah. So only the added fungus, yeah, has any effect. Calm. So you can actually like use it as a control variable, yeah. So that only the added part you can see the effect, not the shit that was already there before, yeah. Standardizes the method, yeah. Part B, yeah. Explain why it was important that the soil contained no mineral ions useful to the plants here. So the reason for that is so that you only see the type of fertilizer that's affecting it, yeah, not the minerals that were there beforehand, yeah. So that's another easy question, yeah. Calm. Question C, yeah. Explain why how groups in A and C act as controls, yeah. So you got in A you got heat treated fungus added, yeah, and nitrate fertilizer, yeah, and then group C you got the same shit but you're using ammonium, yeah. So what it does here is it ensures that the effects of the fungus can be seen, yeah. So what it allows here is that you can see the effect of fungus here, the different types of fungus alone, yeah. So heat treated here, and you can also then therefore see as well the nitrate and ammonium difference here. So you need to have like heat treated nitrate and heat treated ammonia as a control variable for ammonia, control variable for nitrate. You get me? Alright, calm. So D, yeah, is a standard one, bro. They love asking this question, yeah. Suggest what the scientists should have done during the drying process to be sure the water had been removed, yeah. So obviously, bro, standard, yeah. So weigh the samples, yeah, at intervals during drying, yeah, to see if the weight becomes constant, yeah. At E. So what conclusions can be drawn from the data in the table about the following the effects of fungus on growth of the plants here? So in fungus here, it's increasing the growth of the roots and shoots in both of them here. However, yeah, it's going to produce an even bigger growth with nitrate here. So if you look at nitrate, it's gone from um 4 to 9 here. So that's the biggest amount of growth here. But for both shoots and roots here, it's also adding it here. Not 4 to 9, but from 1 to 9 here, even bigger here. Also, you need to state, yeah, that's going to be similar, yeah, for the part B, yeah, it says the effects of nitrate fertilizer and ammonium fertilizer on the growth of the pea, yeah. So obviously there's similar dry masses here for roots and shoots here, and there's no statistical difference here. As the SDs here, the standard deviation here, overlap. So what I'm talking about here is if you look at between the shoots here and the roots here for the standard one, yeah, they're basically the same, yeah. They're not even overlapping, bro. You get it? So the 0.4 and the 0.34, yeah, they're barely overlapping, bro. Same as the other one, the roots and shoots here, their standard deviations are within the same amount, yeah. So that means there's no difference between them and the dry masses here when you got nitrate and ammonium on the normal one, yeah. So when it's just heat treated, so when you got no fungus working, you just got standard, bro. You get me? Nitrate and ammonia. Comp F here. Explain why determination of dry mass was an appropriate method to use in this investigation. Yeah, so obviously dry mass measures the increase in organic material. Yeah, as obviously the water content is gonna vary from time to time. Yeah, so obviously if you have dry mass, it's gonna get rid of that. Yeah. Calm. Final question. Yeah, question G. Yeah. Which treatment gave the best result in commercial terms? Yeah. So obviously fungus with the nitrite. Yeah, gave the largest shoot to root ratio. Yeah. So fungus with nitrate gave the largest shoot to root ratio, yeah, and also the largest dry mass of shoot, yeah, because if you look at the shoot, yeah, it's got 9 grams, yeah, so it's the largest dry mass of shoot, and it's also got the largest ratio too, yeah, so it's working very good, yeah, you get it. Alright, come on, bro, that's the end, fam.